We're going to look at section D.5. We're going to look at the first three assessment statements. First, going to look at the outline of the value of classifying organisms. First thing we want to look at is one of the objectives of classification is to truly represent how living and extinct organisms are connected. First thing we're going to First reason is, first of all, it helps us to identify unknown organism. By knowing how they're all connected, we can use the identification key and use it to identify organisms. A second reason is to see how organisms are related in an evolutionary way. And a third reason is to see a prediction of the characteristics shared by members of a group. Knowing this, like if they have similar blood proteins, we can see how they would react. They should act the same, considering they have the same characteristics. If we know what is in each category, we can tell if they're going to react the same or not. And so that gives us a little insight of how they will react. A fourth one that I would like to add to the text would be it's also for effective communication. All scientists can use the same terminology when they use the taxonomy. Next, we want to look at explaining the biochemical evidence for common ancestry. One thing we know is that every known organism on Earth uses DNA as its main source of genetic information. This is our first evidence of the biochemical, using DNA. This leads to believe that we might have a common ancestor since every living organism uses DNA. In addition, all proteins found in living organisms all use the same 20 amino acids. Not only that, there are right and left-handed amino acids, and an interesting fact is that of all the right and left-handed, they have found only left-handed amino acids in organisms. And then two more I would like to add to this would be that DNA structure is universal, using the same 4 bases. 5, genetic code is universal. In other words, DNA is transcribed to RNA, and it's the RNA that's transcribed to proteins. So all these 5 all suggest a common ancestry, since it's all universal to all living things. Next, we want to look at explain how variations in specific molecules can indicate phylogeny. First of all, we want to look at phylogeny is the study of the evolutionary paths of species. What we're going to look at is the most similar they are, the more closely related, and the higher degree of differences, the less closely related. Kind of makes sense. Traditionally, we've always looked at the morphology, or the physical characteristics, and we said if they tend to look the same, they're going to be related. But with increase in our medical field, we can now look at the molecular differences. Some examples of this is looking at the polypeptide sequences in certain proteins. Some proteins they look at is the hemoglobin and the mitochondrial protein. The closer they are together, the more similar the protein makeup of the hemoglobin and mitochondria, the closer related the organisms are. We can also look at the mitochondrial DNA. And changes in the DNA sequences from genes from one generation to another are due to mutations. The more differences there are between the two species, the less related they are. Here's an example. We have a DNA sequence from species 1. We have a DNA sequence from species 2. They must be very closely related because the DNA tends to be the same. Third species might be a little bit further out. In other words, this would be the oldest, the youngest. This would be the youngest even, with fewer or with little more mutations. And then finally we have this one here. It does resemble this in a way, but it has lots of mutations to it, and so it may not be, it's not closely related, so it could be the same species, just a mutated version of it, or it may not be related at all. So there is a problem when you get too many genetic variations, but you can definitely tell that if they are related, this one is way younger than the other one because it is so far down the line because of the genetic mutations. And how we know this is we can use this as an evolutionary clock. The differences in the polypeptide sequences accumulate steadily and gradually over time. Since it does a steady and gradual rate, we can use this kind of a clock, not to tell exactly what time the mutations occurred, but to tell how closely related the organisms are. So we compare, what we're going to compare is homologous molecules. In other words, molecules that are representing the same from two related species. And it's possible to count the number of places among the molecules where there are differences. The example our book talks about is with species A, B, and C. If species A and C has 83 differences, and A and B only have 26 differences, you know that A and B are more closely related. And that would put A and C further apart from each other in the phylogeny. And also, another the technique they use to do this is type of a DNA hybridization. They can take a homologous pair, so a DNA from one species, the DNA sequencing from the same spot on another species, fuse them together, and they can count how many match up and how many differences there are. The more differences, the less closely related. If we see that there's 83, and there's another neat fact, is that if we have 83 nucleotide differences, it's approximately 3 times more than the 26. So between this A and B, it's 3 times more differences. And so A and C must have become species difference about 3 times further in the past. So the split happened 3 times further for C than it did for B. So it kind of gives an evolutionary time period on how far unrelated the A and C are. And we can look at this one last thing here is looking at the number of DNA sequences. We can also tell how related something is. This is showing that between a human and pig, we are different in only 13 DNA sequences. From us and a duck, 17, and so forth, on down to the moth with 36 differences. So it kind of shows which ones we are closely related to. So we just talked about the first, actually the first 4 assessment statements explaining the value of classifying organisms. We gave 4 values of that. Explaining the biochemical evidence provided by universality of DNA. We gave 5 of that. We talked about how the variations can be used for phylogeny, and we also discussed how the variations can be used as an evolutionary clock.